So I'm well aware that people have spoken of the long period of gaslighting as well as incomprehension that pervades ME and the rather limited research that's gone on around it. Well, I'm here today to tell you about some research we've been doing which really shows biomarkers that have a mechanistic explanation as to what is going on underpinning this syndrome and indeed long COVID-2 and other post-viral diseases. This is a long-standing collaboration I've had with someone in South Africa called Professor Risha Pretorius, and so I acknowledge her. Several of the speakers have pointed out that all of these diseases tend to be post-viral or post-infection diseases. There really isn't any doubt about that when you survey the scientific literature. Sometimes particular viruses have been fingered, such as Epstein-Barr virus. Indeed, very recently it was shown that MS was caused by Epstein-Barr virus. Dengue fever, Ebola, all of these have post-viral syndromes. Rheumatoid arthritis is in fact a post-bacterial syndrome, it's called by Proteus, but many of the sequelae are very similar indeed in all of these diseases, particularly concerning inflammation, and most pertinently and obviously today, ME. And one of the things about these diseases is that they have hundreds of different symptoms, which of course has made it hard for some people to get their head around what the causes could possibly be when there are so many very different syndromes, and whether it's realistic to expect a unitary kind of explanation for something that has so many very different symptoms. But actually, I believe that we have got something close to a unitary explanation for the great majority of these symptoms as a result of our research, which I'm going to explain now. Obviously, post-exertional malaise or post-exertional symptom exacerbation is the hallmark of many of these diseases. But fatigue, which is a more instantaneous thing, and brain fog and various other things, cardio problems, are relatively common in all of the people who have these syndromes. And to explain what this research is, and to give you the explanation as to how it is that it's going to provide an explanation for the basis of these kinds of diseases, I've got to tell you about how proteins fold up. And a famous experiment that got a guy called Christian Andersen a Nobel Prize, the famous experiment is referred to often as unboiling an egg. So what he did, of course, when you boil an egg, it goes into a form that is hard to get back again. He added some substances, urea was one of them, it doesn't really matter, which unfolded a protein called ribonuclease. And then he took them away, and the protein folded back into the form that it started with. That got him his Nobel Prize, and it led to the assumption that not only was it the form it came in, but that was the most stable form. It was a reasonable assumption, totally untestable, because it would have taken the lifetime of the universe to test whether or not it was possibly true. It isn't true. In fact, it wasn't the most stable form, and isn't the most stable form. It was the one it just folded up to quickest. And another obvious example of something like this, which people know about, are prion proteins, in which a particular protein, called PRPC, folds up into a different, usually called rogue form, called PRPSC. The sequence is the same, hasn't changed a bit, but the structure is completely different. It's full of so-called beta sheets. It's extremely resistant to being broken down, so much so that actually you can add an enzyme to proteinase K that breaks down every other protein, leaving this one behind. That is the assay for the prion protein in the rogue form. And these forms are quite common and are generally known with the word amyloid forms. Same sequence, much more stable. And it's a form of self-organization. Now we have to look at how blood clots. My index finger represents the protein fibrinogen. It's the second 91. And the enzyme thrombin is going to come along and nip two little bits of the protein out, and then that protein is going to self-organize onto something that's about this scale, with my finger being a single molecule, and the fiber is going to go that way. And when you look at a lot of them in an electron microscope, they look like a plate of spaghetti, like cooked spaghetti. What we discovered under many circumstances, which are all in the papers, and we'll come to the ME and long COVID in a second, is that these proteins, the blood clotting, can also happen into this anomalous form, which has the same sequence but a different structure, full of these beta sheets, amyloid form, and it looks under the electron microscope like, imagine a plate of spaghetti that you partly boiled and glued together, making an unholy mess that we call dense matter deposits. And what we subsequently discovered was that these two are also this amyloid form, and how we discover it is there's a nice stain that doesn't normally fluoresce unless it's bound to this amyloid form when it fluoresces like stink. So you can't miss it. You look down an ordinary microscope and all the bright green bits are fluorescing because they bound to your anomalous clots, and the rest aren't. And these microclots are between 2 and 200 micrometers in diameter typically. 1,000 micrometers is 1 millimeter, so they're very small. And the consequence of this is what we'll see, going to lead to the causes of long COVID and ME/CFS. We previously observed these clots in a variety of chronic inflammatory diseases, Alzheimer's, Parkinson's, rheumatoid, and then we discovered it, of course, clots are well known in the case of acute COVID, but we discovered also that they're these special microclots that stain with our special stain, it's called bioflavin T, the stain, and then we discovered it in long COVID too, and the number of patients who don't have these, but do have long COVID, is 0. We have not seen anyone with long COVID who does not have these. We call them fibrinoid microclots. They also have hyperactivated platelets, which I'm not going to talk about today. Now a little anatomy lesson on how blood flows around the body. Blood is pumped by the heart, of course, gets oxygenated in the lungs, sets out along the arteries that are thicker, and returns via the veins, and in between the veins and the arteries, with a much bigger surface area because they're much smaller, are the things that exchange oxygen with the tissues, and these are called microcapillaries. And they're typically 4 to 5 microns. In other words, just about small enough or wide enough to let a single blood cell go through. So now you can imagine that if you start blocking these things up with microclots that are resistant to being broken down, just like the prion protein, the blood's not going to get through. The oxygen isn't going to get to the tissues, and therefore you won't be able to do all the things, and Katie mentioned ATP production earlier, you won't be able to make energy aerobically, and you're essentially going to get fatigue almost instantaneously. Depending upon where these blockages occur, that determines which of the symptoms you're going to get, because it determines where you're not getting oxygen into the tissue. So you can have a unitary explanation of these fibrinoid microclots that explains the large multiplicity of symptoms observed in long COVID and ME. So the attraction from a scientific point of view of this hypothesis is it gives a mechanistic explanation of what's going on, and it also then gives you an opportunity to consider how you might treat these things. Additional to the blocking up, which is called ischemia because the blood's not flowing, and that leads to hypoxia, which means not enough oxygen is getting through. Once the tissues have been hypoxic, lacking in oxygen, if you then let the oxygen come back, what happens is something called ischemia reperfusion injury. Normally, oxygen is reduced to water in mitochondria, but if they've been very anaerobic for a while, oxygen can also be reduced after it's reintroduced to half-reduced things, superoxide, peroxide, hydroxyl radical, collectively known as reactive oxygen species. And these things, when you have a great blast of them, will kill your cells, and they will do it with some relatively slow kinetics. So it's why fatigue happens immediately, but post-exertional malaise happens a day or two later, because it takes that long for the cells to die as a result of this ischemia reperfusion injury. So some of the treatments can stop the clots from forming. The doctors don't tend to like that because they're terrified of bleeding problems. But those clotting ability can be measured, so we encourage people to do that. But you can also buy nutraceuticals that get rid of the microclots. Best known one is nattokinase. It comes from the Japanese fermented food natto, which is a fermentation of soybeans by a bacterium called Bacillus subtilis. And that enzyme is not a kinase, it's a protease, and it's called nattokinase. And many people, nattokinase serrapeptase comes from an organism found in silkworms actually. Lumbricase comes from earthworms. These are all available nutraceuticals that many people in the long COVID community have found quite beneficial in alleviating their symptoms because they degrade the clots, as simple as that. So the fact that these things are starting to work and be recognised also gives more evidence for the fact that the microclots are the cause of the problems, because otherwise they wouldn't be doing what. So what do we need next? Well, fairly obviously is the things here to say. We need to invest in scientific research. It is not news that doing science for medical purposes brings cures and indeed benefits the economy as well. So they got a Nobel Prize for establishing that. So it is not news that scientific funding is required, and whether or not under this kleptocracy we're going to see any is questionable. So we have to go and look, probably for the moment, until we get a new government, which at least seems about to happen more quickly than some of us had feared. What we have to do is look at other funders, for instance the Wellcome Trust, which as we mentioned, which can do what it likes. It doesn't have to wait for central spending reviews and how the mechanics of research funding operates in this country. And some years ago I did actually run one of the research councils, so I know how it works. You can't easily target things, but when you see that there's a great necessity, both medically and politically, long COVID is probably the way in. It's not news that long COVID has stimulated research in ME, but by working together, I think there's every chance that we can stimulate funding into this space. And obviously I'm biased because it's our research that discovered these things. We do think that there is now the opportunity to actually find some therapies that get rid of the cause, which we believe are these fibrinoid microclots, to stop the oxygen getting to the tissue. And I think with that I will finish. Thank you for your very kind attention.